For the second part of the video, we're going to take a look at more into the protein structure. So as I was saying and giving you the example of the hemoglobin, okay, it actually was made up of 4 domains. So a domain in a protein means that it's actually a basic structure unit. So, and it is also distinct from those that make up the conformations. This is where, when a domain, is a part of protein that can fold into a stable structure independently. So that means that, for example, in the hemoglobin, even though there are 4 domains, so each and every one of them could fold independently of one another. And different domains can impart different function to protein, and they could also bind to different kinds of substance. So proteins can have one to many domains depending on the protein size. So as you can see, in hemoglobin there are 4, and in some proteins there are more than that. So this is what the domains look like. For example, the carbon structure. And domains usually make up useful proteins, and there are thousands of different combinations of amino acids that could make up proteins, and this would increase if each one had multiple shapes. It actually would also increase the likelihood of being bound to one another. So protein usually have only one useful conformation, because otherwise it would not be efficient, because usually when it has to rotate to conform, it uses a lot of energy. That's why towards the end basically energy is minimized. So if it has more than one conformation, so usually a lot of energy is being wasted on that. So natural selection has eliminated proteins that do not perform a specific function in a cell, and that is how they do. So as I was saying earlier on, proteins are divided further into different families, and in terms of so we're not going to go into details of the protein families, because there are many families involved. So in terms of similarities, they have similarities in amino acid sequences and the 3D structure, and they may have similar functions such as breaking down protein, but of course they would do it differently. As you can see over here, so this is one form of elastase and one form of chymotrypsin. So basically they are a part, or basically a kind of protein that is called a protease. Now, for protein, it has multiple peptides, we know that as much, and we do have some of the non-covalent bonds which form interactions between the individual peptide chain from the 3D structure. And from there, you may have some binding site where the protein interact with either one another or to another substance called a ligand, or a compound, or a subunit, whereby each polypeptide chain of a large protein could basically be connected to a dimer, which means that protein are made up of 2 subunits, because of the di, and it can also be the same subunit, but it could also be from the same or different subunits. So it basically depends. Now, this is basically how a single subunit proteins look like. So you can see that this is our favorite hemoglobin. It has a tetramer structure, and over here we can see that for the hemoglobin it has 2 alpha and 2 beta, and even though from this diagram you may think that they look similar, but in actual they do not. So protein has to be assembled very much like everything else. So protein assemblies usually can form very large assemblies as well. So it can form long chains if the protein has 2 binding sites which are linked together as helix or ring, which will form this elastase protein or elastic protein. So actin is actually one of them. So actin fibers in the muscles and also in the cytoskeleton, for example, is made from thousands of actin molecules as a helical fiber that would strengthen the muscles and the cytoskeleton. So if you look at your muscle, if you have a muscle, everyone has a muscle, I'm kidding. Just that probably if some of you work out or lift weight, you can see more muscles, or your muscle will be more prominent. So, or you can just Google on the difference between muscle and fat. You can see that muscle is denser. So the dense muscle structure is actually because of the actin fiber. Now, protein basically, I've actually touched this in the first video. They are divided into 2 types, globular or fibrous. So a lot of proteins basically are globular in structure. So they have compact shape like a ball with irregular surfaces, and a lot of enzymes are globular. I couldn't think of an enzyme which are fibrous, because a lot of them are actually globular. But the second type is the fibrous protein, usually span a long distance in the cell, kind of like our elastase, or our keratin. So the 3D structure is usually long and right shaped. So probably you can see that a lot of, basically from the previous slides, it says that a lot of enzymes are globular structure, and we know that enzymes play an important role, okay, in bodily function of every organism. So do fibrous proteins have any purpose? Yes, they do. Okay, these are some of the important fibrous proteins. Intermediate filaments of the cytoskeleton, such as the actin fiber, structural scaffold inside the cell, okay, to basically make sure, okay, that your scaffold is strong. Keratin in hair, horns and nails. Now, did you know that you can actually walk on your hair if your hair is actually strong, okay, and it doesn't have brittle or split ends and whatnot, because it actually has a strong structure that is contributed by the fibrous protein. Okay, it also forms extracellular matrix, okay, for example in your bones, binds cells together to make tissues, secreted from cells and assembled in long fibers. And of course, okay, the most famous would be collagen, okay, which is the fiber with glycine ring every third amino acid in the protein, making it supple, okay, or the appearance, okay, of a person with a lot of collagen, okay, to look supple. Or elastin, which is unstructured fiber that gives tissue an elastic characteristics, kind of like, okay, if you ever try, okay, to pull, okay, your hair, if it breaks easily, okay, which means that, okay, you don't really took care, either you don't really take care of your hair, or basically you don't really eat a lot of protein or good protein to maintain the elastin protein in your hair. But if you could just pull it, okay, without breaking it, okay, kind of holds like a rubber band, so that means that is how elastin is, okay, in your hair. So for the collagen and elastin, this is basically how they look like, okay? So they are rod shaped, okay, so they are long in structure, and they are basically formed, okay, from they form a fiber, okay, for example collagen, okay, triple helix. And for the elastin or elastase, okay, so basically this is how they look like, okay, the structure of the protein. All right, now protein have a cross-links, okay, and how do we stabilize, okay, the cross-links, okay? So this is usually done, okay, or occur during oxidoreduction, okay? So, well, again, this is biochemistry. So cross-linkages can be done or can occur, okay, between two parts of a protein or between two subunits. So what happens there, for example, I'm giving here an example of disulfide bond, okay, the SS formed between adjacent SH groups, okay, on the amino acid of cysteine. So that means that, okay, if it forms, okay, the SS bond, okay, or disulfide bridge from previously not a bond at all, so that means that the protein is now have a very strong structure. Regardless of what you do, okay, so you can really break that bond because the disulfide bond is actually the strongest structure. So cross-link, okay, could stabilize some of the bonds in the protein structure. But having said that, it doesn't mean that you could just use cross-link, okay, to stabilize the protein, okay, or to make it basically more flexible or higher stability and whatnot. All right, so I think we will move, okay, to the protein function, and I think in the third video, okay, we will have a lot more, okay, on how the protein could function. So let's start off, okay, with the conformation of a protein, okay. Usually, of course, okay, have or gives its unique function. So protein, when it folds, okay, so it gives, okay, the function, or it contributes to the function. So for a protein to work, it must interact with, also, of course, other molecules, because usually protein doesn't work alone. It works with another protein or with other substances and whatnot. Okay, so most of the time protein would bind, okay, to either another protein or a ligand, okay, or a molecule. Okay, so ligand is the molecule that a protein can bind to. Okay, and of course, okay, you have seen previously in one of the slides, okay, before this, okay, there are binding sites, okay, to a protein structure. So binding site, okay, is a part of protein that interact with the ligand. That is where the ligand would bind to the protein, and together they could either anabolize or catabolize, for example, in the case of an enzyme. So most of the time, okay, it consists of a cavity formed, okay, by a specific arrangement of the amino acids. So again, the structure and how it folds would determine the binding site. All right. Now, I'm actually listing, okay, some of the protein function, not all, because obviously there are too much, okay, but these are some of basically protein function depending on the specific formation. Okay, so have many functions in the cell, kind of like enzymes, or for structure, transport, motor, storage, signaling, receptors, gene regulation, and also special functions. So protein function, again, I would just like to stress this, okay, it depends on the specific conformation. And sometimes, okay, what happens is that, okay, so it has to fold, it has to twist correctly, okay, into a unique shape, okay, before it could function properly. And of course, okay, we have to make sure that the 3D structure, okay, is in great shape, okay, so that it could form that specific conformation. So what happens, okay, is that in almost every case, okay, the function depends on the ability to recognize and also to bind to some other molecule. And usually, like I said, protein does not really work alone, so it has to bind to another protein or another ligand. So one example is antibody. So everyone has antibody, right? So if you remember your biochemistry, I actually could remember biochemistry or general biology. Okay, so when we learn about antibodies, we know that it's actually in Y shape, right? So where does, okay, ligand binds to? And we know that antibodies is a form of a protein. So it basically binds at the Y structure towards the end, okay, of the Y structure, right? The top. Okay. So antibodies, we know, bind to a particular foreign substance, okay, that fit their binding site, and this is usually by an induced fit, which we will see in the third video. So similarly, enzymes recognize and bind to specific substrate. This would facilitate a chemical reaction, okay, for catalysis. So again, okay, another example is our neurotransmitters. Okay, so usually they pass signals from one cell to another by binding to receptor sites on the protein. And of course, okay, this, okay, would make sure, okay, that the membrane of the receiving cell can know that, okay, so we have got this signal, this is what we must do next. So that is how it is done. All right, so that would be the end of our second video. Let's move on to the third video, which basically discusses more on the function of a protein.